I'd like to start by acknowledging that I'm gathered from here at this moment on Worimi land. I pay my respects to the elders past, present and emerging. I'd like to also acknowledge that Callaghan campus is on the land of the Awabakal people of the Pamalong clan. And I'd like to extend my respects to them and the different Aboriginal land that you are all gathered on in different places around mainly New South Wales. And just to note some of them, the Gadigal of the Eora Nation for those who are in Sydney, the Dakinjung, amongst others. So it's very exciting to be doing this. This is part of our Visiting Architects and Experts program, and this is a fantastic program that has been set up at the School of Architecture and the built environment to invite leading thinkers and practitioners to come in and share their expertise and ideas with us and with our teachings. So I'd also like to acknowledge that it's been a very anxiety-filled week with three of our students at the University of Newcastle coming down with COVID. So it's I just want to acknowledge the anxiety and the stress that we're all dealing with. I think as staff members, we're all very magpies are fighting. Yes, honey. We are very cognizant of that, and we need to kind of lean in on each other, support each other. And if there are issues that are arising, please don't hesitate to get in contact with myself and as head of discipline, as well as the kind of coordinator for this subject. I'd also like to say, everyone's been working incredibly hard. They've just completed an amazing, I've already been Darcy and I've been looking at some of the work already, an incredible first 4 weeks of their studies in first year. And so much of this has fallen. Yes, it has, honey. An incredible project, which, it's so exciting to have Helen come in and give this kind of celebration of the end of this first project, which has been centering around the idea of the body and the body as a site and a site of spatial and environmental possibility, and to kind of see it as a research opportunity in which they are actually looking back onto their own bodies in order to kind of mine what their bodies are telling them, in order to kind of think about their own creativity and the possibilities of that creativity. So it's wonderful to have Helen and to have seen some of the work that you've been doing, Helen, in terms of kind of framing that and giving us this kind of rigorous, critical, and also rigorously resolved body of work that is so well aligned with the work that we've been doing over the last 3 weeks. So with that, I'd like to hand it over to do the introduction.
Over to Courtney.
Courtney. Thank you, Courtney. I'm looking at this kind of like sea of faces here.
Awesome. Thank you, Sam. Thank you very much. And I also would like to acknowledge that I am on the land of the Awabakal and Worimi people today, and also pay my respects to elders past, present and emerging. It's great that I have the opportunity to introduce Helen today, but I thought that just before this takes place, I would just give a bit of context to how the lockup is sitting in this great meeting of minds. So the lockup is a contemporary art space in Newcastle, and we are committed to contemporary, experimental and conceptual art practice. So it is really amazing that we have been able to partner with the School of Architecture and Built Environment with this guest lecture, and to have the opportunity to present Helen as one of the artists from the Experimenta exhibition that was on at the lockup until late last week. Sadly, that situation has changed quite quickly, but Helen is here to talk about her work that was included in that exhibition. Just very briefly, just to give a bit of background on Helen. Helen is an artist and researcher based in Sydney and possibly London a while ago. Her practice is something that looks at the ambiguous zones in between different places. In this context, with her work Habitation, it is looking at the perception of the body as a passive recipient of human engineered implants. So I'm very excited to hear about Helen and her practice to date, and so excited that we are able to present this in partnership with the university. So thank you, Helen.
Thank you so much, Courtney and Sam. I'm really delighted to be here, and yeah, it's lovely that this has sort of been an outgrowth of the Experimenta work, the Experimenta Triennial that's on at the lockup at the moment. So to give you some brief background about my practice, I'm an interdisciplinary artist and I'm working in the art science space principally, exploring, as Courtney said, exploring issues relating to the body. So I'm particularly interested in zones that are philosophically or experientially ambiguous. So some of those explorations in recent years have looked at the life-death boundary and how ambiguous that border zone is, and that in fact it's not even a hard and fast border. The intersubjective nature of organ transplants, and as Courtney was just saying, prosthetics, medical prosthetics, and the whole breakdown of the animate-inanimate boundary in relation to prosthetics. So my first training was actually in molecular biology, and I continue to collaborate a lot with scientists and sometimes clinicians. I often need to access specialized experimental protocols and things like that, but I really rely on those interdisciplinary collaborations and really love working that way. So I often do long-term residencies in science institutions, for example, and I also collaborate a lot with members of the broader community who have embodied experiences that connect with the thing that I'm exploring. And in this particular project at the lockup, it's actually myself who is that person, but sometimes it's others. So I'm going to take you through, I'll just go to screen share, through a couple of presentations. Oops. So before I get into the contents, I'd like to acknowledge the Darra people of the Eora Nation, on whose lands I speak from today, and pay my respects to elders past, present and emerging. So I'm calling in from Sydney today. And the first project I'd like to talk about, I'll talk about three main projects in this talk. The first one, The Body is a Big Place, was a collaboration with artist Peter Clancy, and it was an exploration of organ transplantation. And we collaborated with a number of cardiac physiologists and a number of clinicians at the Heart and Lung Transplant Unit at St Vincent's Hospital in Sydney. And we were interested, what sort of got us started in this project was an interest in the intersubjective nature of what did it mean to have somebody else's organ inside your body? What did it mean to have somebody's heart inside your body, for example? And we conceived two different components to this installation. It was a long-term research project as well as an installation. This slide shows one iteration of that. We were interested in actually working with fresh pig hearts and perfusing them so that they were able to beat outside the body of the pig, and I'll tell you a bit more about that. And we also wanted to engage with people who'd had experiences of organ transplantation. So we, this is the one iteration of the heart perfusion setup that we we designed with the collaborating scientists. They are 2 pig hearts that we obtained from abattoirs. So we were quite conscious about the ethics of what we were doing, and very, it was important to us that we weren't sort of killing pigs to realize this project. And the only way we could really get pig hearts that were fresh enough, because we needed to have hearts that were removed from the body of the pig within a few minutes of the death of the pig. So we established collaborations with abattoirs and got access to pig hearts that way. And we had a very defining experience at one of these abattoirs when we were developing the project, where it was on the sort of outer fringe of Sydney, and we were waiting in the abattoir. We had to witness the whole process of animals, pigs being killed in the process of this meat production, which was really confronting. And in one instance, we the guys were sort of opening up the pig for us, and they were giving us the entire sort of viscera and organs of the pig. And on one of these occasions, we had this whole mass of organs sort of sitting on this concrete floor in front of us. There was a huge cacophony of sound going on, all the machinery. We were sort of shouting to be heard by each other. And we were sort of scrabbling through these organs looking for the hearts. And we were really arrested when we saw that the hearts were still beating. So the heart, rather, of this pig, like the organs had been well and truly removed from the body, but the heart was still beating and the intestines were still convulsing. And it was really the first time that I had a deep visceral understanding that death is durational. Death doesn't actually happen in this moment of death that we see on death certificates. Different tissues and different cells close down at different times, at different rates, through different processes. And it was really moving and confronting at the same time to see this little heart still beating. So that sort of brought in this other element into the project of this question about life-death ambiguity, and the kind of slipperiness of that border. In fact, the idea that it's not really a hard and fast border. So this is a still from a heart perfusion performance that we did, and I'll show you some video in a minute, which will show you how that actually operated. Another still of the hearts themselves. In this earlier iteration, we used a blood substitute. So that clear fluid you can see coming out is a physiological buffer, a substitute for blood. In the later iterations that we showed at Science Gallery Dublin and Kapelica in Slovenia, we actually used pig's blood. So blood taken from the same pig that the heart was removed from. And that was a very different kind of spectre. We were also really keen to, as I said earlier, to engage people who'd had experiences of organ transplantation. And we created a video, so underwater video performance work with people who were members of an organ transplant community in Melbourne. So these are people who'd either donated tissue, been organ donor recipients, or were close kin of organ donors. And we were very inspired by a theorist, Margaret Childrick, who's a philosopher of the body, really, and feminist philosopher. And she wrote a really defining paper for us called Contesting Normative Embodiment, Some Reflections on the Psychosocial Significance of Heart Transplant Surgery. And she unpacks all of those sort of elements of transplant surgery that are not really headlined in the transplant scene. So the fact that when you receive an organ, you've actually got the donor's DNA in your body. So you've got their organ in your body, and you've also got their DNA in your body. And Margaret Childrick is trying to unpack that idea that, in fact, she also makes clear that these instances of having two different genomes in your body is actually not that uncommon. Organ transplantation is not the only scenario whereby we can have more than one genome in our body, and this is human genome, not the microbiome. So we, with this swimming pool work, we wanted to create metaphors for that whole experience of what does it mean to have somebody else's organ in your body, but in cases of where the organ donor has died in the process of donating. So that's a common scenario. Somebody's, for example, injured in a car accident and they give permission to donate their organs. So the recipient in normal circumstances will never meet the donor, but usually not know the identity of the donor. So they're given some very basic information like gender and age, but we try to create this environment where we've got a group of people sitting underwater and they're sharing this sort of intimacy, this fluid environment, but normal speech is impossible. They can't communicate with each other. They can't really see each other, and they certainly can't speak to each other or be heard by each other. So part of it was a metaphor for that experience. We spent a lot of time talking to people in the transplant community and tried to find ways to grasp that reality. And other metaphors were also laid in there, like the idea of the meeting room. The participants in this video, as you'll see in the moving image footage, leave and come back to this space repeatedly. This is a still of one of those performers. So I'll show you that video. It's just over 3 minutes. So one of the things that arises out of that reality of the ambiguity of the life-death boundary is really complex ethical questions around at what moment do we decide that it's acceptable to declare death and remove organs from a donor. And there are a whole lot of complexities that arise from that I won't discuss all of them here, but those kind of ethical complexities were things that we were really interested in, and that were offshoots, I guess, or things implied by these phenomena that we're exploring. The next project I'm going to talk about is called The End is a Distant Memory. And it really grew out of that prior project. So in this project, again, well, the starting point for this project was exploring life-death ambiguity, but in a very different context. So it arose out of a conversation I had with Jochen Rink, who is a scientist. At the time, he was at the Max Planck Institute for Molecular Cell Biology and Genetics in Dresden. And we were having a conversation. I gave a talk there, and afterwards, Jochen and I were discussing this idea of, he's a regeneration biologist, so he's very interested in this life-death boundary. And he was speculating that there would be still living cells inside chicken meat that we buy from the supermarket, which that really blew my mind that could be possible. He didn't know that for sure, and I certainly hadn't even thought about that. But I got some funding to do a long-term research residency in his lab, and I did the tissue culture experiments to see whether that was indeed the case. And this image is a shot of the biopsy. So this here is the biopsy where a little piece of thigh meat was taken from the chicken meat. And I was able to extract living chicken fibroblast cells from the chicken meat. And we did time-lapse microscopy on those cells. So I've got some video footage of this as well, but this is 3 stills of a chicken fibroblast cell dividing into 2. So that really blew my mind, that these cells were still viable enough to grow and replicate like this, to be able to be regenerated, to grow and replicate. So it raises all sorts of questions, like, what is the chicken meat really alive? Is it dead? It brings up this whole ambiguity of the subject-object boundary. When the animal is alive, the meat we think of as dead, but if the meat has living cells in it, is the meat actually alive or dead? And that sort of disruption of the living dead boundary, or the life death boundary, and the subject object boundary was really interesting to me. And in the project, I was also very interested in that transition of animal to meat, and then meat going into our bodies. So this is a free range chicken farm in just north of Dresden, in a town north of Dresden. And this is the supermarket where I bought the chicken meat. So the fridge where the gentleman is gazing into is where that chicken meat, where I bought the chicken meat that I did the tissue culture experiments on. And I made a video work, which you'll see extracts from, looking at this transition of a fully feathered bird to denuded bird, and a bird that's almost at the point that we would regard as ready to eat. And I was also thinking here about how when we eat meat, we become part chicken. When we eat chicken, we absorb that meat into our body, and there is a genuine sort of material and literal sense that we become part chicken. And of course, that applies to any food that we're eating. So I was very interested in that blur between animal and human as well. And I worked with the whole, I might explain that in a minute. The other thing I was interested in this project was the ambiguity of the life-death boundary in relation to human near-death experiences. So I'm sure most of you have heard of near-death experiences, where people sometimes retain lucid, sometimes perceptions and sometimes experiences from during the time that they were clinically dead. So we have there's a definition of what clinical death is, and sometimes when people are resuscitated in hospital situations, they're declared clinically dead, but the resuscitation efforts continue, and people can be resuscitated from that point and brought back, and people have sometimes lucid memories. And it's interesting that the prevalence of these near-death experiences, are sort of shorthand NDEs, the prevalence of them is actually becoming greater because we are developing clinical techniques that are getting better at resuscitating people. So there are more people who survive, for example, heart attacks and situations that in the past would have been lethal. So this is a video still, the video is projected onto the floor, as was that previous chicken video I was showing you of the chicken being denuded. And the protagonist, whose body's being sort of moved around, is a man called Rowan Thomas, who had a really very detailed and intriguing near-death experience. I don't have time to unpack the whole content of that experience, but he was a victim of a gun crime in the US when he was traveling there. And in the ICU unit, some days later, he survived the surgery, and some days later he went into cardiac arrest and was declared clinically dead. And during that time, he had a whole series of lucid experiences, including and there are very common tropes with near-death experiences. A lot of people report similar and overlapping types of phenomena. So in his case, he reports seeing his body from the ceiling point of view, looking back down on his body, prone on the bed, with a whole lot of medical paraphernalia coming out of it. He recalls meeting his sister, who was alive, but he recalls meeting her in this whatever space he was in, and he had a number of other vivid images as well. And I actually worked with him as a performer in this work, and he's working, and also 3 professional performers. So 2 of the other bodies in that video are Branch Nebula, Lee Wilson and Mirabelle Wooters, and Alejandro Rolandi is the other performer. So I've got, there's a closer still image, and I'm going to show you some video of that project as well.
So the moving image footage you saw in the latter part of the video, so the Rowan his body being manipulated and the mattress and the sheet falling towards the ground, they were kind of metaphorical treatments of descriptions Rowan gave around his experience. So they were sort of extracts, if you like, and abstracted and made to a degree metaphorical. And in that project, I was very much referencing across the human bodies and the chicken bodies and the sort of pink, fleshy qualities of those bodies and their positioning on the ground and other sort of analogies. So the final project I'll talk about is the project that's currently installed at the lockup, Habitation. And as was said earlier, it was part of the Experimenta. It is part of the Experimenta Life Forms International Triennial of Media Art. And it was very much, in this case, working with my own experience. So I was born with a congenital abnormality in my right hip, which didn't really impede me too much for a long time, but in recent years became really quite debilitating to the point where it was getting hard to walk around and I was needing to use a walking stick and things like that. And in 2019, I had a hip replacement, and I was really interested in working with the bone material that would be excised from my body during that surgery. So I knew that it was very difficult to get permission to work with your own bodily or biomaterial that's surgically excised. And I worked with another artist, Jaden J.A. Hastings, who had done a lot of research around the policy settings and the legal situation for obtaining your own tissue. And through her research, I did get permission to keep this bone material, which is in that specimen jar. It's like my femur head sort of upside down. So the ball of that, the top of your femur bone is that sort of ball that sits in the hip socket. And in this photograph, that ball is sort of sitting upside down. And that darker red bit is a cross section through the top of the shaft of my femur bone. And through one of my sort of guiding theoretical references in this project was Monika Backer's work, who's a Polish philosopher, who has
come up with this idea, lithic intimacies, which is, as she explains it, life's diverse and intimate relationships of exchange and interspecies companionship with minerals. So she's really interested in breaking down the animate-inanimate boundary in relation to living entities and minerals, and all of those sort of collaborative relationships that we have with minerals, and the way life affects minerals, the way minerals affect life. And in my project, I was interested in it from two perspectives. One was the idea that our bones are these largely mineral structures. So calcium forms the sort of dominant material in our bones. So our nature is part mineral always. And obviously calcium is not the only mineral in our body. We have vast amounts of iron in our blood and many other trace minerals. But in this particular project, it was the calcium and the bone I was interested in. And I was also interested in her ideas around the idea of the collaboration that has to go on between living tissue and medical implant for the whole thing to work, for the whole thing to be medically successful. So in one part of the project, I was very keen to make some bone china work using my own bone material and also using animal bone to supplement it. So this is a ceramics kiln with a whole lot of beef bones in it that I'm in the process of calcining, which is heating the bone to 1,000 degrees, burning off all the soft tissue. And then you're left with this really beautiful white chalky material that is just the mineral content, devoid of all the carbon-based soft tissue. And I did that same process with my own bone. So took that through that process and made some bone china sculpture. So this is an installation view of the work that's at the lockup. This was documented down at Plimsoll Gallery in Hobart. And so you'll see some bone-like objects in the foreground and some light boxes in the background, which I'll talk about in a minute. But the ceramic work, the whiter object that you see in the foreground is a bone china rendition of that femur bone that was removed from my body during the surgery. So I was able to obtain the data for the CT scans that were taken of my body prior to the surgery. And the company who did that, the Biomed company, exported the 3D files for me. So I had 3D files of my own bones. And I got 3D prints made of them, made molds of the 3D prints, and then used those molds to cast the clay into. So what you see here is a bone china in the foreground, a bone china femur head that was cut out of my body. And in the background, you see my pelvis and the remainder of my femur bone. And they're rendered in earthenware, so a different treatment to the bone that's taken out of my body. And you'll see that the bone china is a beautiful sort of brilliant white color. The earthenware, that is called a white earthenware, but it's a much creamier sort of color. And that color difference and the difference in the sort of at a close, when you're close to the work, you also see a very different surface texture. The bone china is much finer, and bone china has all of those properties that it's highly prized for. So by using my bone material and this animal bone in the bone china, I really wanted to make manifest our mineral nature. So the fact that our bones can be used to make this very, very hard, tough material, which is bone china. And the bone china is 50% bone. So it's made up of 50% animal bone and then 50% two different types of clay. So I found that like a really interesting sort of material language for exploring this collaboration, if you like, between life and minerals. And the light boxes, I was interested here in coming up with a visual language to look at the transformative process of surgery. So again, these the image on the right is my CT scan augmented with some of my previous x-rays. And then the image on the right is metaphoric treatment around exploring or using a metaphoric language around transformation and change. So that's a close-up of that light box. And there's a detail of it. So the dashed red lines that you see in my bone material is a tracing of where the medical implant will go. So it's got a stem that goes into the femur bone, it's got a cup that goes into the hip cup. And obviously in this image, it's depicted in a sense the midway through the surgery when my bone's been removed, but before the prosthesis has been implanted. And a third component of the project, which is still hoping to do, but it's been very difficult to do it during COVID because of the travel restrictions. But the plan is to do a residency at Symbiotica in the University of Western Australia in Perth, who are a residency that deal with artists who work with living materials, and to actually obtain a human bone cell line. So you can buy human cells commercially. So buy a cell line of human bones and grow it on a small sample of the prosthetic coating. What's really interesting about my particular prosthesis is that it's cementless, which means it doesn't use any glues. And this is quite common now in prosthetic implants used in orthopedic surgery. So what the implant relies on is my own bone cells growing into tiny fissures in the surface of the prosthesis. So it's actually my own cells that glue that prosthesis into my body. And what I find really interesting about that is the whole breakdown of the animate-inanimate boundary that results from that. So not only is there a sort of morphological breakdown, you've got cells growing into little tiny crevices of these implant coatings, but you will also get molecular exchange across that border between cell and prosthesis coating. So there's absolutely both a metaphoric and a literal material breakdown of the animate-inanimate boundary. And it really draws in Monica Backer's idea of lithic intimacy. Lithic, by the way, means, for those of you who don't know, it means stone, things that relate to stone minerals. So you get this lithic intimacy that's being enacted in my own body. I might leave it there, just looking at the time. I'll just escape and stop screen share. Okay.
Wow, thank you so much, Helen. Amazing. We're going to open it up to questions. I thought just before we do, I was a bit discombobulated when we first started. So I did want to just say that it's such a pleasure to be partnering with the Lockup on this talk. I hope we're going to do it again. And I honestly feel that the kind of the commitment the Lockup has to experimental, explorative, and such kind of rigorously critical work makes them such a wonderful partner for what we're trying to do at the University of Newcastle. So definitely want to see this as the first of many. And I also wanted to say, Courtney, even though the exhibition is closed, we can still see quite a lot online. Is that the case?
Yes, we have quite a lot of images and artworks online on social media, Instagram, Facebook. We've just received our professional photographs back today, so we'll be pushing a lot of that material over the next coming weeks as well. So yeah, please engage online as much as you can to see what is a really incredible exhibition.
Absolutely. I think it was such an extraordinary experience to go through it. It's beautifully curated, the work. Each of the pieces is a whole world. So I definitely push everyone here to go if they missed it live to check it out. So I'd like to open up to questions. I have so many questions, Helen, but there's all these amazing people here. I keep thinking, there's this the work is so beautifully and emotively produced through these installations and performances. But I think what is so interesting is this kind of forensic, but also curious encounter with the flesh that you have in this work that really brings to the fore it's this kind of making visible of these kind of central questions, these central ontological questions of who we are and what we are and how we are, like how are we constituted, and these assumptions that I think and we've been exploring it in this studio with students around, what constitutes us as flesh, and all the kind of amazing work that's coming out of particularly feminist philosophers, the post-human at the moment, who are working, of course, so closely with science, and so many of them have come out of science, which is, so interesting, to respond to the ideas of kind of authenticity and truth, that we've kind of held true to the idea about what is it to be a body, and what is it to be a body in the world, and what is it to be a body in this world that we're in now. And I think this, the work, it's all there in the work in such a wonderful way. So I think, Darcy, you're going to be also monitoring some of the questions, but if we open up the chat to anyone who's here, throw it out here.
We've got a raised hand. If you just want to turn on your mic and ask a question, that's great.
Hi, thank you. It feels really archaic to throw up your hand. It feels like being a primary school, but it's also funny. First of all, thank you, Helen, for describing your work so in such a crystal clear way that it feels like you really gave us access in such an interesting way. But also, I just wanted to say that I really love the way you merge the kind of brutal biological truths of the body parts you work with metaphors that just extend it. And from that point of view, I wanted to ask you, why did you use smoke as a metaphorical element in the last work you were talking about? Because in terms of chemical processes, burning, it's a destructive process. And there's no way back. So it's entropic, there's no way back from it. While the process that you are working with is constructive. Cells are multiplying and different materials are fusing. So I'd like to hear more about what you were thinking there. Thank you.
Yeah, thanks for the question. I guess the way I see it is the experience of having the hip surgery was both destructive and constructive. So there is actually a real, as you would know, a real brutality to the process of having the surgery itself. It's literally sort of woodworking tools almost that they're using to cut bone and extract things. And it's actually, I'm not somebody who's at all squeamish, but I haven't been able to bring myself to watch a video of a hip replacement surgery. Just, it's like that's sort of taking me over a line. So partly I would say that the idea of destruction is in there, but there's absolutely the idea of construction as well, and the body's creativity, the body's improvisation, the body's capacity to respond and innovate and improvise in the face of a new situation, a prosthesis arriving. And even in the whole lead up to the demise of my hip joint or the function of it, one of the really revelatory things about having the bone in my hands, like being able to actually handle my own bone material, was there's a massive mushroom sort of overgrowth in the head of the femur. Normally a femur is like a beautiful sort of smooth ball, but in this femur head, there was a huge sort of overgrowth, which was all about the body's adaptation to change and the bone growing that way in order to continue to function as a joint, even if compromised. And that incited a real sense of wonder for me that was there. So in answer to your question, I use smoke in that work as a metaphor for that transformative change, and it involves destruction as well as construction. But there are also some direct plug-in points to the making process of the bone china. So when I did one of those large kiln burns, I did a number of kiln firings of beef bone. I made a huge quantity of clay, even though I only needed a small amount, but to learn the process. And in the big firings, there was a certain point when the kiln reaches a certain temperature, there are clouds of smoke that come out of the kiln. It's kind of horrific. Like, people thought the kiln was on fire. People were actually stressed when they saw the smoke coming out, but actually that was all the soft tissue burning off. 1,000 degrees is about the temperature that we cremate bodies at. So there were these other references laid in there to the bone china making process of cremation, as well as the process of change and transformation.
Thank you. That was brilliant. Thank you.
Pleasure.
I mean, I can maybe have a think about some questions. We've got 5 minutes left. I mean, I'll put in for a question.
There's a question actually in the chat.
I think it's around the idea if maybe it's not really a question, but, and I'm assuming this is true, but maybe let's ask the question. If frozen chicken cells are still viable, perhaps beef, lamb, and other meat have viable cells as well. Did you find that?
So just to clarify, frozen chicken, the cells aren't viable. So if you freeze the meat, that actually kills the cells. So this only applies to fresh meat that hasn't been frozen. But I mean, a lot of the meat we buy isn't frozen. But it would absolutely, in answer to the question, it would absolutely apply to lamb and beef and fish and vegetables, and, you know, any fresh food really is likely to have some living cells still there. If it's frozen, no, that will kill them. But it's extraordinary to think, like, sometimes when I walk around the supermarket, I think about that and I look at all the fresh food and think all of that will still have regeneratable cells in it. I'm just looking in the chat and there are some questions there. Do
you want to, shall I read it out, or do you want me to, or do you want to just answer it?
Yeah, look, maybe read it so that everybody.
Okay, so so how much effort was it to find a medium?
Oh, sorry, there's one earlier than that. I'll read it. I find it fascinating that the pig's heart could keep beating outside the body, such a revelation. When does a person actually die if the cells are still alive weeks after? So, sorry, that was the one that's a comment and not a question. Yeah, that is exactly to the heart of what we were exploring. The fact that when does death happen? It's not, certainly it's not a moment of death as represented on our death certificates. It's definitely a durational process. It's definitely a transitional process. And all of those questions are really difficult to answer because we don't even know for how long cells in our brains might be active. Like, how long does consciousness persist? And that's a question that we don't know the answer to, except we do know that when people are clinically dead, some people come back with clear memories of events, things they saw, things they perceived. So clearly consciousness was still working in those cases. Yeah.
I think I'm feeling like I'm missing some of these questions. So maybe Darcy, I don't know if you can, some of these questions aren't on my chat.
Oh, I'm happy to read them out.
Maybe if you, at least you can see them, Helen.
Yeah. Chloe has asked, how much effort was it to find a medium / conditions to keep the fibroblasts at? Because I know some cells are more fussy than others. Ha ha ha. Yes, this is absolutely true. Fibroblast cells have a reputation for being extra tough cells. They survive beyond the life of some cells. And when we did this experiment, we thought the most likely scenario was that it would be fibroblast cells that we'd find. And that's indeed what happened. In terms of a medium, there are known, in the scientific literature, known protocols for keeping cells alive in culture. The hardest thing was actually to extract the cells and have them still viable at that point. And once you've got them out and they're growing in culture, then it's a much easier process to sustain them. You keep passaging the cells using tissue culture. But it would have been harder, for example, to tissue culture muscle cells, for example. And as you've said, Chloe, there is a very wide span in terms of how robust and durable cells are how long they will survive after death. That's, yeah. And Belle has asked, would love to hear a little bit about how your work and research has affected your own relationship to your bodily experience. Have you been surprised at all by the changes in the way you experience your body and even in the experience of consuming food as a result of your research? Yeah, definitely on multiple levels, that's the case. It certainly made me more, I mean, I was always somebody who actually was very interested in bodies and the experience of being in a body. So that interest, I guess, led to this kind of practice. But it certainly has to deepen my respect for my own body and other bodies. And it's deepened my sense of wonder and reverence and a sense of the unknown. I mean, the human near-death experiences, we have no explanation. We really don't know how, why that happens. There are sort of physiological explanations that are proposed, but they don't explain everything. They could explain that phenomena of the light at the end of the tunnel, the effect on the optic nerve as the brain starts to shut down. They don't explain people meeting dead relatives and people perceiving that things have been said to them. They don't explain people hovering up on a ceiling and actually seeing the medics treating their own body. There's a whole lot of things. And certainly the Habitation, the project, the third project that I described or talked about, that really affected my experience of the hip replacement surgery. I really believe that. It was partly that act of being able to see my bone and hold my bone even. I mean, part of the impulse for that was this idea that I couldn't bear the idea that my bone was going to end up in biohazardous waste. That was just wrong. It felt really important that this bone that had worked so hard for me all my life in these abnormal conditions would be honoured and given some kind of respectful next life stage. So, but on top of that, actually seeing the bone and seeing how misshapen it was compared to a normal hip femur head, that made me, first of all, understand why I needed the surgery. Like, I'd seen x-rays, I'd seen how abnormal the shape of my hip was over time. But when I actually held that three-dimensional object in my hand, I really saw the shape of it. I really understood in my body the shape of it. And I really understood why, how bad the situation had become, why it was so important to have the surgery. And that was somehow a way of helping to process the experience of having the surgery. But it also incited this, like I said earlier, this sense of wonder, like, oh my goodness, look how creative my bone's been. Like, look what my bone's done to deal with this situation. That improvising and that work it did to grow abnormally in order to keep that joint as stable as possible. Just deep respect came out of that for me. So another question from Matthew, did you explore the ethics and consequences of those near-death experiences and the effect it has on the person? Like, are they the same person? Just because their cells are alive, can you still consider them alive? What about brain death? Oh gosh, there are so many layers to that. Where to begin? Yes, we did explore, I did explore in both projects, actually, The Body is a Big Place and The End is a Distant Memory. We explored the ethics of death being durational and a blurred boundary. And we looked at that in the context of medical decisions and medical ethics. So how clinicians make decisions about, because in order to, some of you may be aware that the definition of death actually changed in the late 60s in response to organ transplant surgery becoming possible. So death used to be defined as circulatory death. When the heart stops beating and blood stops circulating, the patient was dead. But they needed to change, something like 10 men in a Harvard medical committee decided to change the definition of death. And it got changed from circulatory death to another nebulous definition, brainstem death or or sometimes whole brain death. So it became defined by the function of the brain, or the brain stem, which is the little bit that sits at the base of the brain that communicates between the brain and the spinal cord. So often when people have catastrophic car accidents and things like that, if their brain stem is badly damaged, they can no longer breathe without being on a ventilator, and they can't, there's very little they can do. So they changed that definition in order to enable organs to be removed from a body of somebody who had this catastrophic brain damage. And that was entirely to enable organ transplants to happen. So already there's a slipperiness about the definition. And even when you break down the definition of brainstem death or whole of brain death, that gets really slippery as well. So you might have a brainstem that is partially damaged. It can't do some things, but it can do other things. Or you might have a brainstem that's damaged, but what if the frontal lobe is fairly intact? Like, could a person still be thinking? Like, is thought still possible in that situation? We don't know that. And there are so many complexities with this newish definition of death that was supposed to clarify things for clinicians. And there is still different mixed feelings within the transplant clinician community about how these questions should be acted on. And it is very difficult I think, for clinicians. I mean, clinicians are dealing in sort of life and death, high pressured situations in a way they need definitions that are as clear as possible to guide their decisions. But actually, when you unpack the situation, there are so many complex shades of grey. So ethics, absolutely. In terms of the effect it has on the person, a lot of people who have near-death experiences do come back changed from the experience. Certainly that was the case with Rowan, the man that I worked with. And in his case, and in a lot of cases, that operates in some ways on a spiritual level, like a changed understanding of their place in the world. Some people have much less fear of death after those experiences, and that's the case with Rowan. So yeah, that's a whole huge lecture in itself, but definitely a lot of effects.
I'm thinking we've probably got a chance for one more.
Feather fellows? Feather, sorry. Which is the next one in the chain. What is the definition of clinical death, and have you formulated your own opinion on the definition or point of death throughout your research? How is that different to the current definition, if it is? In addition, as a scientist, what are your thoughts on the spiritual world? Do you believe in life after death? Yeah, another big set of questions. Clinical death, I would have to review, it's related to how long the heart stops beating, so how long there's no pulse. And I can't actually remember the length of time, but it's a definition that is based on the function of the heart, unlike the clinical definition of the definition of death is related to brain function, but the definition of clinical death is heart function, because that's something that can be monitored by very quickly from the outside. You can feel if somebody's pulse is operating or not. And it's a very slippery question. Like, clinical death is a different definition to death. And clearly people survive clinical death. That's well accepted, even in the medical field. That's a well-described phenomena and well-accepted phenomena. So we know that these definitions are slippery, including clinical death. In terms of my opinion on the definition of the point of death, what I would say about that is that ontologically, death actually defies any clear definition. I think that we have to have working definitions to enable clinicians to make decisions, but I don't think that there's an ontological clear point of death. I think, as I've explored in my work, I think that death is transitional and durational. So death happens over time, and different cells die at different times. And it feels arbitrary to me to pull out one point and say, well, that's death. I think people experientially, like loved ones who are with somebody who's dying, have an experiential understanding of when death happens. And I know that, I've been in that situation. So there's a sort of perceptual sense for people with someone of when death happens. And then there's this medical definition, and then there's this idea that death is actually durational. So all of that sits in there with me, as all of those things are sort of valid ways. But ontologically, to me, there isn't a clear point.
I hate to cut this, but Ethan has such a great question. So maybe if maybe, I mean, I feel like all of these questions need about 20 minutes to answer.
Yeah.
Perhaps a kind of a short answer to Ethan's, because I'd hate to leave that one hanging.
Yeah. So I'll read it out. Most of the amazing arts are combined with attention-grabbing visuals. Is displaying the physical art model alongside with digital visuals the initial and efficient way to explain the idea or meaning with the projects? And how do you decide which visuals are appropriate to display? Okay, so I, as a visual artist, I would think of my physical objects as sculptures rather than models. I do work between two-dimensional and three-dimensional forms. I work between photography and video and as 2D forms, sculpture as a 3D form, installation as a 3D form. And I do move quite fluidly between them, and I pick them up. I use different mediums for different projects. And I do make a call on what seems to be the best or most effective way to convey a certain idea in a certain project. So it's very much case by case. In the case of the living cells or organs, so the pig hearts, for example, it was really important to that project to have the physical hearts there. So the real flesh, beating flesh, little hearts, that was super important. With the cells from the chicken meat, it might have been theoretically possible to grow the cells in a gallery and have people look down microscopes, but that was really logistically so hard. So I made the decision with that one to record them in the science institution and present time-lapse video. Sometimes the work involves live performance, like with the heart perfusion. Sometimes it's video performance, so a performative act is videoed and relayed via moving image. So I really kind of dissect the elements of the artwork. And in the case of my bone, it was really important to me to have the real bone material in there. So to have the real bone china, have that object present in the exhibition as a kind of honoured material of the body. But to show some of those other metaphorical layers, the 2D form working with the extracted CT scans seemed more appropriate. So yeah, it does vary project by project. More, somebody's just mentioned the book, Chloe's mentioned The Immortal Life of Henrietta Lacks. It's a really wonderful read. Thanks for bringing that up, Chloe. Written about a woman called Henrietta Lacks whose cancer cells were removed in the late 1950s, and without her knowledge, without knowledge of her family, and those cells have gone on to become the absolute tissue culture workhouse of the world. There are literally billions of her cells around the world in hundreds of labs, and her story raised all of these ethical questions about how the body should be treated when it's removed from the body. She was an African-American woman. It brought up questions of race and class. It's a really fabulous read.
Okay, I'm going to have to cut it now because I think we've gone over time. But thank you so much for sharing your work with us, Helen, and thank you everybody for participating and coming and engaging. Thank you, Courtney from the Lockup, for partnering with us on this talk. Please have a look at the work if you haven't already, as part of the Experimenta exhibition, and hopefully we can do this again. Thank you, Helen. Stay safe, everybody, and take care.
Thank you so much for having me.